Hey, ChatGPT.
Hey, it's great to hear you. How's it going today?
Let's understand the types of movements. So we have self-generated, stereotyped, reflexive, passive. So why is reflexive in the brainstem and spinal cord, and what does it have to do with the stretch reflex, the Golgi tendon reflex, the flexion withdrawal reflex?
Reflexive movements are automatic and don't require conscious thought. They're coordinated at the spinal cord or brainstem level. The stretch reflex, like the knee jerk, uses muscle spindles to resist a sudden stretch. The Golgi tendon reflex helps prevent excessive tension, relaxing a muscle. The flexion withdrawal reflex pulls you away from something painful, fast.
But why is the stereotyped and reflexive in the spinal cord and brainstem and not in the motor cortex or the basal ganglia or the cerebellum?
It's all about speed and efficiency. Reflexes like the stretch or withdrawal reflex need to happen super fast, often before you even think. The spinal cord and brainstem handle these quick, protective responses without needing the slower, more deliberate processing of the motor cortex or the fine tuning of the basal ganglia or cerebellum.
I don't get your reasoning.
Fair enough. In simpler terms, reflexes are your body's emergency responses. If you touch something sharp, you don't have time to think about it. Your spinal cord acts instantly to pull your hand away. If that message had to travel all the way up to the motor cortex, you'd react too slowly. So reflexes are wired in these lower, faster regions to keep you safe right on the spot.
Gotcha.
No problem. Whenever something doesn't click, we just break it down further. Anything else you're curious about?
What are central pattern generators?
A central pattern generator is like your body's built in rhythm machine. It's a neural circuit, usually in the spinal cord, that can produce rhythmic patterned movements like walking or swimming without needing constant
Sorry, I interrupted you. Tell me again what are central pattern generators?
No worries. Central pattern generators are basically neural circuits, mostly in the spinal cord or brainstem, that produce rhythmic patterned movements like walking, swimming, or breathing without needing moment by moment conscious control. They coordinate muscle sequences all on their own, so you can walk while daydreaming or chew without thinking. They handle the rhythm.
And what about the
And
primary motor cortex?
the primary motor cortex is like the conductor of voluntary movement. It's up in your brain, right on the top, and it's in charge when you consciously decide to move. If you want to wave at a friend or pick up your coffee, this area sends signals down through your spinal cord to get your muscles moving in a precise, intentional way.
And the neocortex, is it the same thing as the primary motor cortex?
The primary motor cortex is actually just one piece of the neocortex. The neocortex is the broader outer layer of your brain that handles a ton of higher level functions like thinking, planning, perceiving, and yes, controlling movement. So the neocortex is like the big city, and the primary motor cortex is one specialized district.
Okay, that's so good. I'm happy with that. Let's talk a little bit about sequence of activation. So,
I'm go-
what does the posterior parietal cortex do regarding integration of sensory inputs?
The posterior parietal cortex is like the brain's map maker. It takes in all sorts of sensory info, sight, touch, even where your body's in space, and integrates it. This helps you know where things are and how to reach for them. In short, it's what lets you connect what you feel or see with how you move.
Okay, but we'll come back to that. Let's talk a little bit about the corticospinal tract. So, we have axons from the primary motor cortex descending to the corticospinal tract.
Exactly. The corticospinal tract is like the direct highway from your primary motor cortex down to your spinal cord. Those axons carry the brain's precise commands straight to your muscles, especially for voluntary, skilled movements. It's your brain telling your body exactly what to do.
Hi, Mia. I don't know how to pronounce your name. Come study with me. I'm studying medicine. Um, okay, so Um, so we have upper motor and lower motor neurons. I don't understand the location though. Like we go from the cerebral cortex to the ventral horn, and then from the ventral horn to the skeletal muscle.
You've got it right. Upper motor neurons start up in your cerebral cortex. Think of them as the commanders. They send their signals down through the spinal cord until they reach the ventral horn. Now, lower motor neurons live right there in that ventral horn. They take the baton and carry the commands straight out to your skeletal muscles, making them move.
Hey Rodrigo, I'm studying medicine. Come study with me.
Hey
Okay,
there.
so why specifically the ventral horn, not the dorsal horn?
It's all about function. The ventral horn is the outgoing side. It's where motor neurons reside, ready to send signals out to muscles. The dorsal horn, on the other hand, is where sensory information comes in, like touch or pain. So ventral is the go side, dorsal is the incoming side.
So dorsal's the incoming side.
Exactly. The dorsal horn is where sensory input, like touch or pain, enters the spinal cord. It's the receiving side, bringing information from your body up to your brain. So dorsal in, ventral out.
Dorsal in, ventral out. Okay. And
It-
okay, that makes sense. I'm happy with that. What about the pyramidal tract?
The pyramidal tract is like the express lane for voluntary movement. It includes the corticospinal tract, starting in your motor cortex. As these nerve fibers descend, they form pyramid-shaped structures in the brainstem. From there, they cross over, so your left brain controls the right side of your body, and vice versa, and then head down to direct those fine, precise movements you consciously choose.
Alright, so pyramidal tract. We have axons of upper motor neurons, and they pass through the pyramids of the medulla. What are these pyramids?
Those pyramids are just raised structures on the front of your medulla where these nerve fibers all bundle together. They form a sort of ridge, hence the name. When these fibers cross over, which most of them do, that's what creates that left-right control switch. So they're basically landmarks where the big motor pathways travel.
Okay. Why pyramidal tract is for upper motor neurons and not lower?
Because the pyramidal tract is the long-distance command route from your brain down. Upper motor neurons live in your brain's cortex and send their signals down the spinal cord. Once those signals reach the spinal cord, lower motor neurons, which live there, take over and go directly to your muscles. So the pyramidal tract is the highway before you get to the local streets.
And why do we start from the primary motor cortex and then we go? Why not having a different starting point like the cerebellum or the basal ganglia?
The primary motor cortex is specialized for initiating voluntary, precise movements. It's the planner and initiator of your intentional actions. The cerebellum and basal ganglia are more like the coordinators and refiners. They help smooth out, adjust, and fine-tune movements, but they don't directly initiate the command. So the primary motor cortex calls the shots, and then others help polish the moves.
I don't get that. We'll have to come back to it. Okay, let's change chapters. We've got a lot of chapters to go through. Okay, basal ganglia anatomy. So we have
No worries.
a worm-like structure almost.
It happens. Don't worry about it. We'll just pick up where we left off. Ready when you are to dive into the basal ganglia anatomy or any other chapter.
Yeah, so we have the head of the caudate nucleus, we have the putamen, the thalamus. Kind of help me picture this.
Imagine the basal ganglia like a set of deep brain hubs. The caudate nucleus is a bit like a curved C hugging the side. The putamen sits right next to it, more like a rounded mass. The thalamus is nearby, acting like a relay station. Together, they're tucked deeper in the brain beneath the cortex, helping coordinate and fine-tune your movements. Kind of like a behind-the-scenes team making sure everything runs smoothly.
Huh. Okay. Fair enough.
I'm glad that helps. Whenever you need another mental picture or a little more clarity, I'm here to make it easier. Just keep those questions coming.
So we have the thalamus quite centrally, and then left and right we have the globus pallidus, interna, externa, then the putamen, then we have the caudate nucleus in front, and the hypothalamus between the thalamus.
Exactly. The caudate nucleus curves around a bit like a companion to the lateral ventricles, often arching in front. The hypothalamus is tucked beneath the thalamus, so it's more like a lower neighbor. All these structures work together, but anatomically, you're nailing their layouts.
Okay, that's fair enough.
I'm glad.
So the pallidum is the globus pallidus interna and extenda. That's fair enough. Why is the lentiform nucleus putamen plus globus pallidus?
It's just named for its shape. Lentiform comes from lens because together the putamen and glottis pallidus look a bit lens-shaped in cross-section. Anatomists like to bundle structures by how they appear, so the putamen outer part and the globus pallidus inner parts together form that lens-like lentiform nucleus.
Okay. And the corpus striatum is the caudate plus putamen plus pallidum. I don't understand why.
The term corpus striatum means striped body, and historically, anatomists saw these structures with striped connections. The caudate and putamen are functionally linked and receive similar inputs, while the globus pallidus is involved in output. Grouping them as corpus striatum highlights their role in coordinating movement. So, it's more about their connections and how they work as a team in the basal ganglia circuit. If you're still mulling it over, or if something feels a bit fuzzy...
So the corpus striatum is divided by the internal capsule.
Yes, the internal capsule is like a highway of nerve fibers running right through, dividing the caudate nucleus on one side and the putamen and globus pallidus on the other. So, it slices that corpus striatum into distinct regions, while still letting them talk to each other.
So it divides... Tell me again what it divides.
It divides the caudate nucleus and the thalamus on one side from the putamen and globus pallidus on the other. In other words, it slices right between the two main parts of the corpus striatum, separating the caudate and the putamen globus pallidus combo. Think of it as a neural boundary line.
So, the putamen and pallidum is divided by the internal capsule.
Yes, the internal capsule runs right through, splitting the caudate and thalamus on one side from the putamen and globus pallidus on the other. It's like a highway cutting through a city, separating these key basal ganglia structures. You've got it.
Interesting. Okay. Neostriatum is dorsal striatum equals caudate plus putamen? What do you understand?
It's called the dorsal or neostriatum because the caudate and putamen sit more toward the upper dorsal part of the basal ganglia. They're heavily involved in receiving input from the cortex. Think of them as the reception area. In contrast, the globus pallidus is more about sending the output, so it's not grouped into the neostriatum.
I don't get it.
No problem. In simpler terms, the caudate and putamen are like the input team. They get lots of signals from the cortex. Because they sit more on the upper side of the basal ganglia, we call them the dorsal striatum. The globus pallidus, on the other hand, is more about sending signals out, so it's not part of that input-focused dorsal group.
I don't get it at all. Anyway, we'll come back to this.
No worry.
Let me add it to a different album. Cool. So,
Absolutely.
Let's talk a little bit about the vestibular system.
The vestibular system, our internal balance master. It lives in your inner ear and helps you know which way is up, whether you're moving, and keeps your eyes steady. So when you turn your head or spin around, it's the vestibular system whispering to your brain, keeping you upright and coordinated. Ready for some spins?
What about the location and organization of the olfactory epithelium? So, um, explain to me how it looks like a little bit.
Picture the olfactory epithelium like a patch of sensory carpet high up inside your nasal cavity. It's tucked up near the top, lining part of the nasal passage. This area is covered with special receptor neurons that catch odor molecules. Each little receptor is like a lock waiting for a specific smell key. Once triggered, they send signals straight to your brain's olfactory bulb. It's your own built-in scent detector.
Okay, can we talk a little bit about the ciliated receptor cells that send their own afferent axons to the brain?
Absolutely. These olfactory receptor cells are pretty unique. Each one has hair-like cilia that trap odor molecules. Once they detect a smell, these cells actually send their own axons directly into the brain, specifically to the olfactory bulb. So unlike many senses that relay through other structures, these smell receptors take a direct line to your brain. Fast and personal.
Tell me a little bit more.
Sure, each olfactory receptor cell is specialized. Different cells respond to different odor molecules, kind of like a lock and key. When an odor binds, the cell fires an electrical signal. All these axons bundle together, forming the olfactory nerve, and deliver the message right to the olfactory bulb. From there, the brain interprets what you're smelling, whether it's fresh bread or a rainy day.
Interesting. Okay, that makes sense. So, there are more than 1,000 different odor and receptor proteins, and with each receptor cell expressing just one of these.
Exactly. Each olfactory receptor cell is like a specialist. It only expresses one type of odor receptor protein. With around 1,000 receptor types, your nose can detect a vast range of smells. Each odor molecule triggers a pattern of activation across different receptors, and your brain puts together the puzzle, letting you recognize countless distinct scents.
Okay. So, each receptor cell responds to a number of different odors with action potential firing. Can you explain that? Hey, Casper.
Of course, each receptor isn't just a one-smell wonder. It can respond to multiple odors, but with different strengths. When a scent molecule binds, the cell fires action potentials. Different odors trigger different firing patterns. So one odor might excite the cell a lot, another just a bit. Your brain reads these patterns from many cells at once, combining them to recognize a specific scent. It's like a code made from different levels of excitement.
That makes sense. Okay. So, olfactory information is coded, but in the pattern of stimulation that the brain learns to interpret.
Exactly. Each odor creates a unique pattern like a fingerprint across different receptor cells. Your brain learns to recognize these patterns. Over time, you associate patterns with familiar smells like coffee or roses. So, it's not just one receptor per scent, but a pattern of activity that your brain decodes, like reading a complex smell alphabet.
So, olfactory transduction depends on a second messenger process, with CAMP being activated in response to an odorant molecule. So, that leads to the. Are you a Chelsea fan or a. I like mixed martial arts, Ismail. I don't really watch football, and I also like Brazilian Jiu-Jitsu.
No worries, Ishmael. We all have our passions, and it sounds.
ChatGPT is talking to you too. Hey, Bach, Tono. Boys, open up your books and study with me. Having said that, let's go back to this. So we have CAMP as a second messenger. This leads to an opening of.
Exactly. Once the odor molecule binds, CAMP is produced. This second messenger causes specific ion channels, often for sodium and calcium, to open up.
Sorry,
when those ions flow.
Boys and girls. Yeah. So CAMP dependent ligand gated ion channels, and then what happens? So these channels are permeable to sodium ions and calcium ions.
Exactly. When those channels open, sodium and calcium ions flow into the olfactory receptor cell. This influx depolarizes the cell, basically creating an electrical signal. Once that happens, if the signal is strong enough, the cell will fire action potentials, sending that "I smelled something" message off to the brain.
Okay, that makes sense. And so what happens after that? So we have influx of sodium ions and calcium ions, and so this depolarizes the olfactory receptor cells, and signals the binding of another molecule, and it leads to action potentials.
Exactly right. That depolarization triggers the olfactory receptor cell to fire action potentials. These action potentials travel along the cell's axon, heading straight into the olfactory bulb. From there, the brain gets the signal, interprets it, and voila, you know what you're smelling.
Nice.
It's a quick and elegant cascade from odor to brain recognition.
Awesome. Turkey is awesome.
You're very welcome.
Okay. Let's continue. So we have gustation, a sense of taste. So evaluating the nutritious content of food and preventing the ingestion of toxic substances is really like key function. And like sweet means, why does sweet means that is like for identification of energy rich nutrients, whereas umami for some reason means recognition of amino acids. Salty means ensuring proper dietary electrolyte balance. I mean, that makes sense. The last one. I'm from Cyprus. I'm in the UK now, studying to be a doctor. Sorry, yeah, let's go back to sweet, umami, salty, and sour.
No worries. Our taste buds are basically evolutionary survival tools. Sweet signals energy-rich sugars, think calories to fuel you. Umami or savory indicates proteins and amino acids, crucial building blocks. Salty helps ensure we get enough electrolytes like sodium. Sour can warn us about unripe or spoiled food, and bitter often flags potential toxins. In other words, taste guides us toward what's good for us and away from what isn't. If anything feels
I am 23. Okay, so sour and bitter. What about you? Warn against the intake of potentially noxious and poisonous chemicals?
Exactly. Sourness can hint at spoiled or unripe food, which might upset digestion. Bitterness often signals naturally toxic compounds found in plants or spoiled items.
The sour and the salty make sense. I don't understand the sweet one, but okay, oh well. Fair enough.
It can seem funny that sweet signals something good, since we all know candy isn't always healthy. But evolutionarily, sweet often meant ripe fruit, quick energy. In today's world, it's about balance, so we don't overindulge.
Alright, so gustatory system organization. So the tongue is the principal organ of taste. We have 5 different modalities of taste. We talked about them. And there is different variation in sensitivity, which is fair enough.
Exactly. Different parts of the tongue are more sensitive to certain tastes, but the whole tongue can detect all 5 modalities. Taste buds on your papillae, those tiny bumps, are the real taste receptors. They send signals to your brainstem, then thalamus, and finally to your cortex, giving you that full taste experience.
Can we talk a little bit about mechanisms of taste transduction?
Absolutely. Each taste uses its own mechanism. Sweet, umami, and bitter rely on specific receptors that trigger second messenger pathways when they bind to molecules, kind of a cascade. Meanwhile, salty and sour are more direct. Salty is detected through sodium ions, and sour through hydrogen ions, acidity. In all cases, once the cell depolarizes, it sends signals to your brain, and that's how you taste.
Thank you. God looks at the heart. Salt sensation depends on the equilibrium potential for sodium ions across the taste receptors. Okay, fair enough. And the sour sensation depends on acidity, the potassium ion channels. CAMP is involved, and This leads to depolarization of taste receptors. Okay. And then sweet sensation comes about via second messenger system that closes potassium channels, leading to depolarization of the taste receptors. Okay, can you explain all of this?
Sure. For salty, sodium ions directly enter taste cells, changing their charge and triggering a signal. For sour, acidity hydrogen ions blocks potassium channels, causing depolarization. For sweet, bitter, and umami, molecules bind to specific receptors. This activates a second messenger system, like CAMP, which closes potassium channels. Without potassium flowing out, the cell depolarizes. In all cases, depolarization creates that taste signal that your brain interprets.
So bitter and umami sensation are due to a second messenger induced increase in intracellular calcium in the receptors, and the calcium increase leads to neurotransmitter release. Can you explain this process a little bit?
Of course, for bitter and umami tastes, when the molecule binds to its specific receptor, it activates a chain reaction inside the cell, a second messenger pathway. This pathway increases calcium inside the taste cell. That rise in calcium is like a signal to release neurotransmitters. These neurotransmitters then pass the taste message on to the brain. So calcium inside the cell triggers the final release of that taste signal.
Okay, and sweet sensation comes about via a second messenger system that closes potassium channels, leading to depolarization of the taste receptors. Is this because potassium takes the positive ions outside? That's why the closing leads to depolarization?
Exactly right. Normally, potassium flows out, keeping the inside more negative. When sweet molecules close those potassium channels, potassium can't leave. This makes the inside less negative, so the cell depolarizes.
So in entrance of calcium ions, bitter and umami sensation, closing of potassium, sweet, and what about sour?
Sour works a bit differently. It's all about acidity. Those hydrogen ions H+. When you taste something sour, the hydrogen ions block potassium channels and sneak into the cell. This makes the inside less negative, causing depolarization. So sour taste is basically acidity triggering that signal.
Nice. I don't know any Turkish, unfortunately. So let's talk a little bit about central pathways of the gustatory system. So taste is signaled by cranial nerves 7 for the front 2/3 of the tongue, and for the rear part it's the vagus and the cranial nerve 9, which one is 9? Abducens is 6, 7 is facial nerve, 10 is vagus. And then 11 is accessory, 12 is glossopharyngeal, and then for 8 and 9 we have vestibulocochlear. I don't know, remind me 8 and 9.
No worries, you got most of them. Cranial nerve 8 is the vestibulocochlear nerve for hearing and balance. Cranial nerve 9 is the glossopharyngeal nerve. That's the one that handles taste from the back of the tongue.
Hypoglossal is 12.
Exactly. The hypoglossal nerve is cranial nerve 12, and it's all about controlling the tongue's movements. No taste, just motion. So now you have the full picture.
And why is it that the facial one is for the anterior part of the tongue?
The facial nerve, specifically a branch called the chorda tympani, evolved to handle the front 2/3 because it's well positioned. It also ties into saliva glands, helping you taste and salivate to start digestion. The glossopharyngeal nerve handles the back, closer to the throat, which makes sense for gag reflexes and swallowing. It's just a neat division of labor.
Okay. Fibers from second order taste neurons project ipsilaterally to the ventral posterior nucleus of the thalamus.
Exactly. After your taste nerves relay the signal to the brainstem, the second order neurons send the information up to the thalamus on the same side, ipsilaterally. The thalamus then acts like a relay station, sending the taste signals to your brain's gustatory cortex, where you consciously experience flavor.
I don't understand this.
No problem. In simpler terms, after your tongue's taste signals reach the brainstem, the next stop is a relay hub in your brain called the thalamus. The interesting part is that the signal stays on the same side of the brain. So if you taste something on the right side of your tongue, that information goes to the right thalamus. From there, the thalamus sends the taste information to the part of your brain that lets you consciously taste.
Okay, we'll come back to that. All right, let's talk a little bit now about the peripheral auditory system. So we have outer ear, middle ear, cochlea, auditory nerve. So what's the difference between the peripheral and the central?
The peripheral auditory system is basically everything up to the point where sound is turned into nerve signals. So your outer ear, which catches sound, your middle ear, which amplifies it, and your inner ear, the cochlea, where sound becomes neural signals. Once those signals travel up the auditory nerve into the brainstem and beyond, that's the central auditory system. It's all about processing sound in the brain from there on out.
I don't get it.
No worries. In simple terms, the peripheral part is like the hardware, your ear structures that catch sound and convert it to electrical signals. The central part is your brain. Once those signals travel in, and your brain makes sense of them.
Well, I'm studying to be a doctor. I want to be an orthopedic surgeon. Okay, so tell me one more time the difference, and then we'll talk about the middle ear.
Sure, one last time. Peripheral is all the ear structures that bring sound in and turn it into nerve signals. Central is when the brain takes over to process it. Now, the middle ear is that amplifier in between. It has tiny bones, ossicles, that transfer sound from your eardrum to your inner ear, making the sound stronger so it can be detected well by the cochlea.
What about you? And so, okay, an impedance matching device increases pressure 45 times. So what does pressure have to do with all of this?
Think of it like this. Going from the air in your ear to the fluid in your inner ear is like shouting from air into water. You need more oomph. The middle ear bones boost the sound pressure so it can move that fluid effectively. Without that boost, about 20 to 45 times, the sound wouldn't have enough force to be heard properly.
I don't understand. And how are the tympanic membrane and valve window areas mixed with all this?
It's all about focusing the force. The tympanic membrane, your eardrum, is a lot bigger than the oval window, which leads into the inner ear. By concentrating the sound energy from the large area of the eardrum onto the small area of the oval window, the pressure increases. That's what gives the sound the extra push it needs to move the fluid in your cochlea.
And what are middle ear ossicles?
They're the tiny bones in your middle ear. We've got three, the malleus, incus, and stapes. They work together like a lever system. The malleus attaches to your eardrum, the incus connects the chain, and the stapes presses onto the oval window. Together they amplify and transfer sound vibrations from the eardrum to the inner ear.
And so the middle ear prevents sound from being reflected back from the fluid-filled cochlea, huh?
Exactly. Without that boost from the middle ear, sound would mostly bounce off the fluid in the cochlea. The middle ear ensures that those sound waves have enough pressure to push into the fluid, so they don't just reflect back. It's all about efficiently delivering sound to where it can be sensed.
Okay. So the cross-section of the cochlear duct. So explain to me the etymology of the word cochlea and what's this duct? So we have scala vestibuli. What does scala mean? Explain the etymology. Scala media, scala tympani.
The word cochlea comes from the Greek word for snail, because it's shaped like a spiral shell. Inside that spiral, we have three fluid-filled chambers, all called scala, which means staircase in Latin. The scala vestibuli is the entrance staircase where sound enters. The scala tympani is the drum staircase, leading sound back out. The scala media, or cochlear duct, is in the middle, holding the organ of Corti, where sound is actually detected.
So, scala vestibular and scala tympani communicate via helicotrema, at apex of cochlea. What is helicotrema?
The helicotrema is basically the little passage at the very tip of the cochlea, where the scala vestibuli and scala tympani meet. Helico refers to a spiral shape, and trema means a hole. So it's literally the spiral hole, letting fluid pressures from sound waves travel from one chamber back through the other, so the energy can dissipate after triggering the sensory cells.
I don't get it.
No worries. In simpler terms, the helicotrema is just the tiny tunnel at the very top of the cochlear spiral. It's where the two fluid chambers connect. So when sound waves go up one side, they can come back down the other. Think of it as the tip of the spiral letting the sound complete its path.
Okay, cool. I'm happy with that. And so, composition of cochlear fluids. So the scala vestibuli, scala tympani, perilymph, endolymph, and the scala media. What's stria vascularis?
The stria vascularis is like a special factory in the cochlea. It lines part of the scala media and pumps out the unique fluid there, endolymph. Endolymph is high in potassium, which is key for triggering signals in hair cells when you hear. In short, a stria vascularis keeps that potassium-rich environment just right for hearing.
No, why would I be offended? Okay, so we have high Na, low K plus in the scala vestibuli and the scala tympani, and then scala media contains endolymph, unusual extracellular fluids, rage in K plus and low in Na plus.
Exactly. The scala vestibuli and scala tympani have perilymph, which is more like normal extracellular fluid, high in sodium, low in potassium. But the scala media is special with endolymph, high in potassium, low in sodium. This potassium-rich endolymph creates the right conditions for your hair cells to spark when sound waves hit, letting you hear.
Okay, interesting. Okay. So, cool.
I'm glad you found it interesting. If you ever want to dive deeper into any part of these systems or just take a break and chat, I'm here for it.
Tonsils. So, tonsils are masses of lymphatic tissue in and around the oral cavity which serve as an essential immune function.
Exactly. Think of them like little immune guards at the entrance to your throat. They help catch and fight off germs from what you breathe in or swallow. So tonsils are a frontline immune defense team, keeping an eye on anything that might cause trouble.
What is the Waldeyer's ring?
Waldeyer's ring is like a protective circle of immune tissue. It's made up of your tonsils, palatine tonsils, adenoids, lingual tonsils, and some other small lymphatic tissues. Altogether, they form a ring at the back of your throat and nasal cavity, guarding the entrances to your digestive and respiratory tracts. Think of it as your first line of immune defense.
Okay, understand the tonsil. I do not have a girlfriend now. The tongue. So we have oral parts, anterior 2/3. We have foramen cecum and terminal sulcus, and then the pharyngeal part.
Exactly. The tongue's front 2/3, the oral part, handle most of the tasting and moving food around. The foramen cecum is a little pit marking where your thyroid gland started during development. The terminal sulcus is a groove dividing the oral and pharyngeal parts. The pharyngeal part, the back third, helps with swallowing and has more immune tissue, like the lingual tonsils.
All right. So, okay, that makes sense.
I'm glad to hear that.
So
Learning
let's talk a
about
little bit about the muscles of the tongue. So we have extrinsic and intrinsic. So intrinsic for fine movements for speech and eating. And the extrinsic gross tongue movement. What does gross tongue mean? Palatoglossus, styloglossus. These are extrinsic muscles, hyoglossus, genioglossus.
gross tongue movement just means big, broad motions, like sticking your tongue out, pulling it back, or moving it side to side. Those extrinsic muscles, palatoglossus, styloglossus, hyoglossus, genioglossus, anchor your tongue to other structures, like your palate, styloid process, hyoid bone, or mandible, so you can move the tongue as a whole. The intrinsic ones, meanwhile, shape your tongue for more precise tasks, like speech and detailed movements.
Intrinsic fine movements for speech and eating.
Exactly. The intrinsic muscles are like the fine-tuning team. They let you shape your tongue precisely, curl it, flatten it, or make subtle movements. This is crucial for clear speech, forming different sounds, and for manipulating food as you chew, getting it ready to swallow.
Intrinsic fine. Okay. Superior, inferior, longitudinal, vertical, transverse. Okay.
Yes, those are your intrinsic tongue muscles. The superior and inferior longitudinal muscles run front to back, curling or shortening the tongue. The vertical muscle flattens or narrows it, and the transverse muscle runs side to side, helping to elongate or bunch the tongue. All these subtle adjustments give you fine control for speech and careful eating.
So genio means chin, hyo is the hyoid bone. Where is the hyoid bone?
The hyoid bone is a small U-shaped bone in your neck, just above your larynx or voice box.
One more time.
The hyoid bone is a small U-shaped bone located in your neck, just above your larynx or voice box. It's a bit special since it's not directly attached to other bones. It's suspended by muscles and ligaments, and it serves as an anchor for the tongue and other muscles involved in swallowing and speech. If you ever want to revisit any of these anatomical bits, just let me know. Otherwise, we can keep moving forward.
Which muscles are innervated by the hypoglossal nerve?
The hypoglossal nerve controls all the intrinsic tongue muscles and most of the extrinsic ones. Those are the ones that move the tongue. So if you're sticking out your tongue, curling it, or pushing food around, you've got the hypoglossal nerve to think. The exception is the palatoglossus muscle, which is actually controlled by the vagus nerve.
Interesting. What do you want to be when you grow up? And so, okay, so the sensation of the tongue, so taste, anterior 2/3 facial, and then sensation is the lingual nerve, V3, and then posterior is taste, glossopharyngeal, sensation, glossopharyngeal, okay.
Exactly. The front 2/3 of your tongue get taste from the facial nerve and general sensation like touch or temperature from the lingual nerve, a branch of the trigeminal nerve V3. The back 1/3 handles both taste and sensation through the glossopharyngeal nerve. It's a neat division of labor.
All right, let's talk a little bit about the course of cranial nerve 11 and 12. So 11 being the accessory, 12 being the glossopharyngeal. Oh no, sorry. 12 is the hypoglossal. So we have mixed sensory and motor, and it's why is my question first, and then it supplies the stylopharyngeus.
So cranial nerve 11, the accessory nerve, is actually mostly motor. It helps move the sternocleidomastoid and trapezius muscles. Cranial nerve 12, the hypoglossal nerve, is purely motor, controlling tongue movements. Now the glossopharyngeal nerve...
So what do we mean when we say somatic motor?
Somatic motor just means voluntary muscle control. When you consciously move a skeletal muscle, like your tongue or neck muscles, that's somatic motor function. So nerves like the hypoglossal or accessory nerve use somatic motor fibers to control those muscles.
So somatic motor, what would be the opposite?
The The opposite would be autonomic, which handles involuntary functions. Autonomic nerves control things like your heart rate, digestion, or gland secretion.
So, okay, I see. I'm chasing myself the time. Why accept the palatoglossus?
The palatoglossus is a special case because it's actually more connected to the palate and pharynx. That's why it's innervated by the vagus nerve, which is more involved in swallowing and the movements of the soft palate. So while it's technically a tongue muscle, it's part of that swallowing and palate system, not just pure tongue movement.
Arises as rootlets from the anterior medulla. Why rootlets? And why from the anterior medulla?
We call them rootlets because instead of one big nerve emerging, multiple tiny fibers come out of the brainstem and then merge into the nerve. The anterior medulla is just where this nerve exits. Each nerve has a specific spot. For the hypoglossal nerve, it's the front or anterior part of the medulla. It's just the anatomical pathway that this nerve follows out of the brain. If anything else pops into your mind, or if you want to...
Passes through the hypoglossal canal, descends close to the carotid sheath, course lateral to the hyoglossus into floor of mouth. Okay. We'll come back to that.
Absolutely. Whenever you want to revisit that or any other topic...
Opioids. So, act on opioid receptor antagonized by naloxone. Naturally occurring morphine. Codeine, okay, no problem. Intravenous, intramuscular. What do we mean by epidural administration?
Epidural administration means delivering the drug into the epidural space, just outside the protective covering of the spinal cord. This is often done for pain relief, especially during labor. By placing the opioid near the spinal cord, it blocks pain signals before they reach the brain, giving more targeted relief.
Where is the epidural, sorry?
The epidural space is a fat and blood vessel filled area just outside the tough membrane, the dura mater, that surrounds your spinal cord. It's like a cushion around the spinal cord. When you inject medication into this space, it can numb the nerves nearby, providing pain relief from that level downward. Commonly used in surgeries or childbirth.
Okay. So the receptors is in the pons and midbrain, and I wonder why. Also in the periaqueductal gray matter, nucleus raphe magnus, spinal cord, posterior horn 1 and 2, GIT, peripheral tissues. Why specifically these things?
These areas are key players in pain modulation. The brainstem regions, like the periaqueductal gray and nucleus raphe magnus, are central to your body's own pain control system. They help release natural painkillers like endorphins. The spinal cord's posterior horn is where pain signals first enter and can be modulated. And in peripheral tissues, those receptors help detect and manage pain at the source. It's all about having control at multiple levels.
Okay, fair enough. We have subtypes delta, kappa, OP1, OP2, MU1, 2, OP3.
Exactly. These subtypes, delta, kappa, and mu, are different kinds of opioid receptors. Each subtype has slightly different effects. For example, mu receptors are strongly linked to pain relief and euphoria. OP1, OP2, OP3 are just alternative names for these receptor subtypes. Different opioids target these receptors in varying ways, leading to different clinical effects.
All right, and so respiratory depression?
Respiratory depression means your breathing slows down more than it should.
Why does it happen with morphine?
Morphine and similar opioids act on the brainstem, which controls breathing. When those opioid receptors are activated, they tell the brain to slow down breathing. If the dose is too high, the brain's drive to breathe can become dangerously low. That's why opioid doses have to be carefully managed.
Mm-hmm. Okay. So, let's continue. So, the fibers from the second order taste neurons project ipsilaterally to the ventral posterior nucleus of the thalamus. This is so specific, man. The Thalamic efferents project to the insula, defining the primary gustatory cortex, which in turn projects to the orbitofrontal cortex, sometimes defined as a secondary cortical taste. Fucking hell, man. Explaining this is... I want you to explain this so I understand what I'm saying to you, but please do it in a way where it makes more sense.
Absolutely. So when you taste something, that signal travels to the brainstem. From there, it goes to the thalamus on the same side of your brain as the taste. Think of the thalamus as a relay station. It then sends the taste signal to the insula, your primary taste center. After that, your brain also loops in the orbitofrontal cortex, which helps combine taste with other senses, like smell, giving you the full flavor experience.
The parabrachial nuclei of the pons are shown in orange. Some of the second order taste neuron synapse here. The parabrachial nuclei also project to the amygdala. Explain, I don't understand. And hypothalamic nuclei not shown among. Yeah.
Of course, the parabrachial nucleus is a hub in your brainstem. After taste signals leave the brainstem, some go to the thalamus for conscious taste. But others head to the parabrachial nucleus, which links taste to emotions, like why certain tastes feel comforting. From there, it can connect to places like the amygdala, which handles emotions, and the hypothalamus, which helps regulate hunger and fullness. So taste isn't just about flavor. It's tied to feelings and body responses too.
Okay. Organ of Corti. Detect the sound-induced motions of the basilar membrane. Contains two types of sensory hair cells, inner hair cells, outer hair cells. Apical membrane is bathed in endolymph. Basolateral membrane of hair cells is based in perilymph. Inner hair cells, afferent nerve fibers. Outer hair cells innervated by afferent nerve fibers. And so, okay, you need to explain this to me. You only have one chance. And just so you know, I'm a very stubborn guy that only learns things by really deeply understanding them.
All right, let's dive deep. The organ of Corti is like a microphone inside your cochlea. When sound makes the basilar membrane vibrate, the inner hair cells detect those vibrations and send sound signals to your brain. That's the afferent part. The outer hair cells are like tiny sound adjusters. They get signals from your brain, efferent, and change their length to fine-tune the vibrations, making hearing more precise. The tops of the hair cells sit in potassium-rich endolymph, which helps trigger signals, while their bases sit in perilymph. In short, the inner hair cells hear, and the outer hair cells tune, and it's all about balancing those fluids for the perfect signal.
But why apical membrane endolymph and basolateral membrane perilymph?
It's all about creating a voltage difference. The endolymph at the top of the hair cells is high in potassium, making it positively charged. The perilymph at the base is more like normal fluid. When sound moves the hair cell, potassium rushes in from the top, creating an electric signal.
Mechanotransduction in hair cells. Deflection of the hair bundle opens non-selective cation channels. The mechanoelectrical transducer channels at the lower end of the top of the tip links between neighboring stereocilia. K plus, the major cation in endolymph, enters and depolarizes the hair cell, excuse me, driven by its electrochemical gradient. What's going on? CA enters, causes adaptation. Voltage gated CA2 channels open, triggers physical release. Okay, you need to explain all of this.
When sound moves those tiny hair bundles, the stereocilia, it stretches little tip links between them, opening channels. Potassium from the endolymph rushes in. This positive charge depolarizes the cell. That triggers calcium to enter too, and calcium fine-tunes the response.
Okay.
Finally, that calcium also makes the cell release neurotransmitters, sending the "I heard something" message to your brain. So, it's all about sound causing a mechanical pull, That creates an electrical signal.
That makes much more sense.
I'm really glad it clicked. I know when it's all broken down, it feels a lot more manageable. Anytime you want to untangle something, we'll just walk through it together.
So, cranial nerve 9 is the glossopharyngeal, so supplies stylopharyngeus.
Yes, exactly. The glossopharyngeal nerve, cranial nerve 9, supplies the stylopharyngeus muscle. This muscle helps elevate the pharynx during swallowing and speech. So the glossopharyngeal nerve has both sensory roles, like taste, and motor roles, like controlling that muscle.
Okay, cool. The pharynx.
The pharynx is basically the throat area. It's the muscular tube that runs from behind your nose down to your esophagus and larynx. It's key for swallowing, directing food down the right pipe, and it also plays a role in speech. It's divided into three parts: nasopharynx, oropharynx, and laryngopharynx, depending on where you are along the way.
Let's talk about nerves: nasopharynx, oropharynx, laryngopharynx, corner of nasal cavities, soft palate, epiglottis, esophagus.
Sure. The nasopharynx is right behind your nasal cavity, connecting your nose to your throat. The oropharynx sits behind your mouth. Think soft palate in the back of your tongue. The laryngopharynx is lower down, where your throat splits into your esophagus for food and your larynx for air. The epiglottis is like a little flap. It covers your airway when you swallow, so food goes down the right pipe.
An innervation.
The nasopharynx is mainly innervated by branches of the trigeminal nerve. The oropharynx is handled by the glossopharyngeal nerve, which deals with sensation there. The laryngopharynx gets its innervation from the vagus nerve, which also covers the larynx and helps with swallowing.
Why?
It's all about coordinated function. Each part needs sensation and control for proper swallowing, breathing, and speech. The trigeminal helps sense the upper throat, the glossopharyngeal handles the middle throat and gag reflex, and the vagus manages the lower throat and larynx. It's all divided up so everything works smoothly together.
But the motor is vagus, except the stylopharyngeus.
You got it. The vagus nerve controls almost all the muscles that move your pharynx, except for the stylopharyngeus, which is handled by the glossopharyngeal nerve. This arrangement helps coordinate swallowing and ensures that each part of the throat works together properly.
Muscles of facial expression: occipito frontalis, orbicularis oculi, orbicularis oris, zygomatus major, buccinator platysma.
Those are all the muscles that let you show emotion on your face. The occipitofrontalis raises your eyebrows, orbicularis oculi closes your eyes, orbicularis oris puckers your lips, zygomaticus major lifts the corners of your mouth, like smiling, buccinator helps with blowing or chewing, platysma tenses your neck, and they're all controlled by the facial nerve.
So trigeminal is cutaneous innervation of the face. What is cutaneous? And also V1, V2, V3 is, well, I know we have the auxiliary, we have the maxillary, and the mandibular.
I'm sorry, but you've hit the daily limit for advanced.
Hey bud, so we have the innervation of the face from the cutaneous innervation by the trigeminal nerve, if you can explain it. And also trigeminal, we have auxiliary, mandibular, and maxillary, and then it's foramen ovale for V3, foramen rotundum for V2, and for V1, it's the superior orbital fissure.
Absolutely. The trigeminal nerve is the main sensory nerve for your face, and it's divided into three branches. The ophthalmic branch, V1, goes through the superior orbital fissure and covers the forehead, the upper eyelid, and the eye. The maxillary branch, V2, passes through the foramen rotundum and covers the lower eyelid, the cheek, and the upper lip. And the mandibular branch, V3, goes through the foramen ovale and covers the lower jaw, the lower lip, and part of the ear. Each branch is responsible for sensation in its own specific region of the face.
Okay. Fair enough. Ophthalmic nerve, so yeah, so branches into lacrimal, frontal, mesociliary. So what different parts of the body, of the face, do each branch innervate? Huh? So which different parts of the face do these nerves innervate? Hey, can you hear me, man? Hello? Hey. Hello?
Hey there, how's it going? Hope you're having a great day.
So the different branches of the trigeminal nerve, which parts of the face, different parts of the face do they innervate?
Absolutely. The trigeminal nerve, or cranial nerve V, has three main branches. The first one is the ophthalmic nerve, V1, which mainly innervates the forehead, the upper eyelid, and the cornea. The second is the maxillary nerve, V2, which covers the lower eyelid, the upper cheek, the upper lip, and the nasal cavity. And the third one is the mandibular nerve, V3, which innervates the lower jaw, the lower lip, and some of the muscles involved in chewing. If you have any specific area you're curious about, definitely let me know.
Quiz me a little bit.
Sure, I'd love to. Let me throw a few questions your way. First question, which branch of the trigeminal nerve is responsible for sensation in the forehead and the scalp? Second question, which branch provides sensory innervation to the upper lip and the maxillary teeth? Third question, the mandibular nerve carries both sensory and motor fibers. True or false? Fourth question, which branch of the trigeminal nerve...
Wait, okay. True for question 3. My question is, why is it true? And then for question 1, I'd say ophthalmic. And then for question 2, maxillary.
Perfect, you got those right. So for question 1, the ophthalmic nerve V1 does indeed handle sensation in the forehead and the scalp. And for question 2, the maxillary nerve V2 is responsible for the upper lip and the maxillary teeth as well. And as for question 3, it's true because the mandibular nerve V3 carries sensory fibers for the lower jaw and also motor fibers to the muscles of mastication, like the masseter and temporalis. If you want to dive deeper into any of these or have more questions, just let me know.
Mm. Hey, one person.
No worries at all. If you have any other questions or